Okay, guys, hope all is well. Quick one today, just to set the stage here for gel electrophoresis. I would imagine most of us could guess at what electro means, something to do with electricity, something to do with charges. Phorese means to carry. And so what we're going to see is that somehow electricity carries DNA in a gel, much like a jello. So today we'll do a virtual lab, and then you'll also maybe see a demonstration of this in a real gel electrophoresis chamber. If we were at school, we'd be doing this in lab, probably this week. So our goal is, can we explain the process of gel electrophoresis? But before we do that, just quickly, some review here of what we learned about genetic engineering so far. Remember, this kind of old school genetic engineering, selective breeding, when you're trying to get some desirable traits from a couple of organisms together, right, in one organism by breeding them. There's two different types of it, so see if you can remember what type is shown here. Okay, so this is inbreeding. Remember, that's when you have very similar organisms, right? Purebreds, and you're breeding those together to create an offspring, right? So you're trying to sort of exacerbate the traits that you already like, right? To try and maintain that line. Good things about that, but also some bad things too. Another type of selective breeding is hybridization. And remember, that's when you take things that are a little bit different and you like some traits here and you like some traits here, and you put those things together and you get an offspring, and you hope that that offspring has both the traits that you liked in the two things that you bred together. Okay, yesterday we started talking a little bit about the four tools of genetic engineering. So now we're talking about sort of the biotechnology in vitro form of genetic engineering. And there's some things you need in your toolbox to be able to do this. So if you remember, you need something to cut DNA. That's your restriction enzymes. You need to be able to make copies of the DNA once you've cut it out of a chromosome or cut a gene out of a bacteria or wherever you got it from, a virus. You need to be able to make copies of it. That's polymerase chain reaction, or PCR. Then you need to be able to separate it. How do you separate all these copies of DNA that you made? And that's really what we're going to be getting into gel electrophoresis today. Ultimately, once you've separated it out, you might want to take some of those pieces of DNA and recombine them with other pieces of DNA, and that's what we're going to work on later in the week, recombinant DNA. But those are really the four steps of biotechnology if you want to try and create a recombinant organism. Okay, so just quickly here, remind ourselves, restriction enzymes, right, cut, and they cut in a specific way. And you really want to use restriction enzymes that produce sticky ends. So remember, these restriction enzymes are naturally occurring in bacteria. They're kind of the immune system of bacteria in a way, because they cut up foreign DNA, but they'll cut any DNA if you put them in contact with the DNA. So if you have a bunch of DNA, human DNA, viral DNA, whatever, and you want to cut it into pieces so you can analyze it further, restriction enzymes are what you're going to use. We'll talk about some of the more specific forms of it and some of the better forms of it a little later in the week. You maybe have heard of CRISPR-Cas9. That's one of the newest sort of ways of cutting DNA very specifically. So we'll get into that later. And then remember, polymerase chain reaction or thermal cycling is really taking now this piece of DNA that you've cut out. You've got only one piece. That's not enough, right? So you cut out a gene from an organism, or you cut out a section of DNA. You're not really sure if it's a gene or what it does yet, but you need more of it so you can analyze it. PCR is that copy machine. So it's really a matter of heating the DNA up, breaking the hydrogen bonds between the nucleotides, and having some polymerase, an enzyme in there to help rebuild your two complementary sides there. And you keep going through that process until you build literally billions of copies of DNA. Okay, so now you've got billions of copies of DNA. And they might all be different lengths, right? Because you just cut up a chromosome into a bunch of pieces with restriction enzymes. You made a bunch of copies of those pieces, but those pieces might all be different lengths. How do you separate them? That's what we're going to look at today with gel electrophoresis. So this is really a means to help us read and analyze DNA. And as I indicated, you start with a gel, sort of as it says in the name, and it's essentially looks like Jello. It's very similar to that stuff we used for our cell size lab, that kind of jello-y auger type stuff. And you put some little wells in it. Imagine sort of a comb that you'd use to comb your hair stuck in this stuff, right? You know, you got little holes in it on one end, and you put drops of your DNA in it, right? DNA that's been added to some restriction enzymes, and it's already been copied. Now you want to separate those lengths of DNA out so you can look at them individually and figure out, are they a gene? What do they do for me? That type of thing. So that's what this does for us. DNA is negatively charged, which, because it's a charged particle, it's allowed to be carried using electricity. So essentially now you'll put that gel in a chamber. You will add electricity to it. There'll be a fluid, a buffer in there that also carries electricity, carries a current. And what will happen is the DNA that's sitting in those little wells will start to move out of those, migrate out of those wells, based on the fact that it has a charge and based on the fact that it's different sizes. Okay, so the pieces will move out, and if they're smaller pieces, they'll move out further, faster. If they're larger pieces, they don't get out of the well, quite as quickly. And we'll learn about that in the virtual lab today. So after the notes here, check out the virtual lab and sort of the videos to explain gel electrophoresis. Again, I hope all things are well as they can be. Take care.